Yeah. Oh, sorry about it. So these receptors represent the largest CCR families, and they are characterized by the classical 7 transmembrane architecture with 7 transmembrane helices and 3 extracellular loops and 3 intracellular loops with an NNC terminus as well. Apart from that, class A GPCRs are they also have some conserved motifs present in them. And here I'm showing an overlaid structures of the beta-2-AR receptor in both active and inactive conformation. The most important conserved residues observed in these receptors are the CWXP motif, the PIF motif, DRY, and the NPXXY as well. And all these motifs are important for regulating receptor activation. So I'm showing all those motifs present in the beta-2-AR structure. And in class A GPCRs, another conserved site is present for sodium binding that is involved for maintaining the inactive state of the receptor. So for my PhD project, I am interested in a specific type of class A GPCR, which is the vasopressin receptor family. This receptor family has 3 different subtypes, V1AR, V2R, and V1DR. V1AR couples to GQ subunit of G protein and mediates calcium mobilization and is expressed throughout the body and is involved with V1AR is involved with multiple physiological processes, including cardiovascular system and some neurophysiological conditions as well. While V2R is quite location specific and it couples to G-alpha S and mediates cyclic AMP signaling and is mostly responsible for fluid homeostasis. V1BR also couples to G-alpha Q like V1AR. However, it is quite location specific when it comes to expression, only found in the pituitary gland and is responsible for hormone release. Now, these receptors are looking at their importance and involvement in physiology. They are quite important from a therapeutic perspective. Now, all of these receptors are activated by a single peptide ligand, which is known as the arginine vasopressin. It is non-peptide, and I have divided the structure of AVP in 2 different parts. The cyclic ring at the bottom that is formed by cysteine 1 and 6, and the C terminus that is extended by the other 3 amino acids. Now, under normal conditions, AVP mediates different physiological functions via vasopressin receptors, as I already mentioned in the previous slide. However, when the AVP levels are abnormally elevated, that can cause severe physiological conditions, including kidney disease, congestive heart failure, and certain behavioral conditions such as autism. Therefore, several antagonists are being developed to target these receptors throughout the past years. Here, I'm showing a handful of vasopressin receptor selective and non-selective compounds, and these are all small molecule non-peptide compounds that were developed to target either V2R or V1AR or both of them in the hope of treating these different clinical conditions. Even though vasopressin receptors are so important and they are pharmacologically well studied, and including both from the agonist and antagonist side, but the structural information remains quite poorly understood. The reason I'm saying this, when I started this PhD, only 4 structures of B2R were available, all of them captured at active conformation, 3 G-protein coupled structure and 1 low resolution Erskine coupled structure. At that time, there were no inactive state structures existed for V1AR, no structural information was there, neither active nor inactive, which posed a big knowledge gap in understanding how ligand modulates these receptors' activation and inhibition. With that, I have, to address this gap, my PhD focused on the following research aims, which is to determination of antagonist-bound inactive state V1AR structure, and then determination of AVP-bound active state V1AR structure, and as well as the inactive Witwer structure determination as well. Together, these aims were designed to provide a comprehensive structural understanding of vasopressin receptor, both activation and inhibition. Now, to begin with, we focused on determining the inactive state structures first, as we were interested in studying the antagonist binding. Now, as we all know, to determine GPCR structures, methods such as X-ray crystallography or electron microscopy are used. Now, for electron microscopy to be able to successfully determine a GPCR structure, the complex needs to be a large, big one. Now, for active site structure, it's easy because the presence of G protein heterotrimer makes the complex bigger, and if the complex can be easily visualized by Cryo-EM. However, this is not the case with inactive or apo-GPCRs. And it's even more difficult for class A receptors, specifically because these receptors lack a sizable extracellular domain outside the receptor micelle. And inherently, they are quite smaller compared to other receptors. So without anything hanging outside the receptor micelle, it's really difficult to determine the structures of class A GPCRs at inactive states using Cryo-EM. Nevertheless, in the past year, several strategies have been developed to stabilize these receptors at inactive states, and most of these strategies include replacing the dynamic ICL3 with more rigid proteins such as Brill, calcineurin, or PGS fusion. However, there is one problem though. All of these strategies, they are not readily reproducible or transferable to other receptor systems, and also extensive receptor engineering is required. Until recently, a camelid antibody referred to as nanobody 6 have been reported to couple to kappa-opioid receptor ICL3 at an inactive conformation. And interestingly, nanobody 6 can bind to the kappa-opioid receptor ICL3 even when this loop is grafted onto other receptors. And this was quite interesting when this paper came out in 2022. So this approach eliminates the need of extensive receptor engineering, which was quite common in previously existed methods such as Brill or other fusion strategies. So in my PhD project, we decided to go with this approach to determine the vasopressin receptor structures at inactive states. Now, to implement this strategy, I defined a set of sub-aims. First, to design the receptor constructs using AlphaFold, then validate them in pharmacology-based assays, and finally to determine their structures at inactive states. And so now I'll walk you through this pipeline, starting with the construct design strategy. For this approach to work, as we know that we need to insert the kappa-opioid receptor ICL3 into my V1AR. However, there's a problem. Even though this approach was published to be the universal one, it is not. We don't know what is the optimum length of kappa-opioid ICL3 required for maximum nanobody 6 engagement. And it might vary from receptor to receptor because not every receptor has the same length of ICL3 or TM5 or TM6. Therefore, we chose 3 different points of fusion based on sequence conservation and sequence alignment. And with different combinations, we generated 9 different chimeras of V1AR carrying different length of the kappa-opioid receptor ICL3. Now that we have all these chimeras in hand, we don't know by inserting this loop if we have introduced any helix break or the TM domain is intact or not. In order to understand that, we took the help of AI, where I used AlphaFold to predict the structures of all these different 9 chimeras. Now I'll walk you through the color scheme. If we go from blue to red, it's an increasing PLDDT score, which means if we see a blue region or a green, AlphaFold thinks that that region is not stable enough or quite flexible, and it's not a good confidence. AlphaFold is not quite confident in predicting that region. When compared to red region, AlphaFold thinks that it's quite stable and it is confident. So if we look at these different constructs, and I have highlighted the fusion region where the fusion is, and we can see all these constructs were predicted with varying confidence scores. If we look at this chimera 4, the loop is predicted with a higher confidence score compared to Chimera 5. Further, the TN7 was also predicted with difference confidence score. This was really interesting to us because we have not made any changes to the TN7 as well. Now that we have all these constructs in our hand, we wanted to test them in pharmacology for their ability to bind nanobody 6 or not. So we took all these 9 chimeras to pharmacology. And this brings me to my second survey. Yeah. So we developed a bread-based assay to study this construct, where the receptor is tagged with allottate and nanobody 6 is tagged with Venus. In the absence of an agonist, the receptor will be at an inactive conformation, which will favor the binding of nanobody 6 to the receptor, which can be observed with increased breadth. Now, when agonist is added, the receptor will no longer be at an inactive conformation. It will attain an active conformation, which will no longer favor the binding of nanobody 6, leading to its dissociation from the receptor, and we can see a decrease in breadth. So the take home from this is that if we see a decrease in breadth upon agonist stimulation, we can say that when there was no agonist, nanobody 6 was bound to the receptor. And we tested all our 9 different chimeras. However, only 3 of them showed robust nanobody 6 engagement. Here I'm showing the top panel shows the kinetic traces of nanobody 6 dissociation from the receptor when increasing concentration of agonist was added. And the bottom panel shows the area under the curve. And I'm also showing the AlphaFold predictions side by side. Only 3 of the highlighted chimeras out of the 9 gave us nanobody 6 engagement. Then we wanted to observe if what will happen if we had antagonists to our assay, in our assays. Can antagonists still bind to these designed chimeras? For that, we use the same method, with an only difference of incubating the cells expressing the receptor with antagonists prior to breadth measurements. And it was hypothesized that when these receptors will be exposed to the antagonist, they will be at an inactive conformation, and that should increase the breadth. Here I'm showing, and a single concentration of AVP was added to observe the competition binding. Here I'm showing us on the green graph here shows the kinetic trace of AVP induced nanobody 6 dissociation, and the top right curve there graph here shows the inhibition of nanobody 6 dissociation from the receptor with increasing antagonist concentration. We tested, first we tested the ones V1R selective antagonists that I already mentioned previously. And we have got, with increasing antagonist concentration, we have got inhibition of nanobody 6 dissociation from the receptor. And with maximum antagonist concentration, we got complete inhibition of nanobody 6 dissociation. This suggests that these antagonists bind to the receptor and makes it in an inactive conformation and prevents the dissociation of nanobody 6 that was previously observed in response to AVP. And we tested another compound, which is a non-selective V1R antagonist, conifaptan, and we have got similar results with increasing antagonist concentration. Nanobody 6 dissociation is inhibited. So we performed these assays multiple times, and we found out that constructs having higher basal breadth have got higher AVP-induced breadth as well. And interestingly, they were predicted with a higher confidence score. So I want to draw your attention to this chimera 1 and 4. Chimera 1 and 4 were predicted with a higher basal, showed a higher basal breadth in the absence of agonists, suggesting more nanobody 6 binding. Again, these 2 chimeras showed higher AVP-induced breadth in response to AVP, again suggesting more nanobody 6 engagement compared to chimera 2. Now, if we look at these AlphaFold predictions, chimera 1 and 4 were predicted with a higher confidence score compared to chimera 2. And this together suggests that the constructs with higher predicted structural stability should also show improved functional behavior. And based on these results, we chose chimera 1 and 4 for structural studies. And then we took the constructs, chimera 1 and 4, that showed us better nanobody 6 engagement to structure determination. And I'm just briefly skimming through this methodology, and Alok has already quite extensively explained it. So we expressed and purified our protein from insect cells, took it to our size exclusion chromatography, where it is separated based on size. And we know our sample complex size, so we would take that sample elution and put it to our microscope to get a structure. Then, so before going into the complex bound structures, we were interested in understanding how this receptor and antibody 6 complex, or how to study this receptor under ligand-free conditions. So I purified both the chimera 1 and 4 under ligand-free conditions, and we can see here in the SSC trace, I have got two populations. And when I ran those samples on gel, we can see a dimer and monomer population for peak 1 and peak 2. Now, peak 1 and the gel picture is not quite visible here. The peak 1 and peak 2, both of them have got both the dimer and monomer bands. However, if you look closely, peak 1 has intense dimer band, where peak 2 has an intense monomer band, suggesting the predominant species in both these peaks. Nevertheless, if we look closely, V1 chimera 1, the amplitude of the peaks are quite small compared to chimera 4, as well as the nanobody 6 band, which is present in both the peaks for both the constructs, is quite visible in chimera 4, not in chimera 1, again suggesting that there might be some stability issue here. That is, V1 chimera 4 nanobody 6 complex might be more stable compared to chimera 1. We took both these monomer and dimer samples to Cryo-EM. However, due to a resolution limit, we couldn't form stable 3D reconstructions for the monomer. And therefore, here I'm showing the Cryo-EM data from the dimer samples. These are the 2D classes that we got for the dimer samples for chimera 1 and chimera 4. In both cases, we can see nanobody 6 bound to the receptor in the micelle with both the protomers, and nanobody 6 bound to both the protomers. In chimera 1, the classes looked quite blurry, and the secondary structural features are not quite visible. Also, one very important point, in chimera 1, nanobody 6 is not bound to both the protomers in most of the classes. Whereas if we look at the chimera 4, most of the classes, the 2D classes, the secondary structural features are quite crisp and evident. And most of the classes also retain both the nanobody 6 bound to both the protomers as well. Then however, we couldn't get a high resolution 3D reconstructions for either of the samples, even though we could see the TM domain and nanobody 6 bound to both. And as I already mentioned, for chimera 1, only one protomer had the nanobody 6 density, whereas in chimera 4, we had the nanobody 6 density in both of them. This again suggests that chimera 4 might be more stable compared to chimera 1. And based on these results, chimera 4 was chosen to move ahead with for further structural characterization of V1AR and not the chimera 1. Then once we purified and tested our samples under ligand-free conditions, we used the antagonists to stabilize the complex further, and I purified this complex in presence of the two compounds that we already tested in pharmacology, one selective and one non-selective compound. And we have got similar biochemical profile, and the gel, chromacy gel here, we can see both the dimer, monomer, and the nanobody 6 band present in both the samples, suggesting the successful complex formation. Then I took these monomer samples first to Cryo-EM. And here I'm showing the vasopressin and quantiferin bound monomers. And in the 2D classes for both the samples, we can see the receptor in the micelle and nanobody 6 bound to it. And in the 3D reconstruction as well, we can see the TM domain and nanobody 6 density as well. And interestingly, we could also see a little bit of helical density. But if I know this is low resolution, but for TM, for vasopressin bound monomer, we could see something coming up or protruding at the top of the binding pocket, which could belong to ECL2, but we couldn't see something like that. Nothing was protruding up in our quantiferin bound monomer structure. We couldn't get a high resolution structure for the monomer samples, maybe because the samples are the complex size is quite small and the data set were quite heterogeneous as well. Nevertheless, we moved to screening our dimer samples. And we have got the Vasopressin-bound dimer structure at 2.8 resolution. In this dimer structure of V1AR, we can see both the photomers clearly bound to nanobody 6 and ECL2 lifting upwards. And another interesting point is that we can see that these two dimers are surrounded by certain lipid-like densities. Now, these lipids could play a role in stabilizing this receptor at this conformation, or these lipids might come from the membrane or the co-purified lipid micelles. And we can also see the ligand present in the two protomers. And here I'm just showing a side view of how the ligand is occupied in the binding pocket. Given the resolution, we successfully modeled V1AR into that density with the ligand. And here I'm showing the binding pocket where Vasopressin interacts with multiple residues in the binding pocket. The hydrogen bonds are shown as cyan, and the white dotted bonds are nonpolar interactions. Now, to validate these interactions, we tested them in mutation assays. On the left here, I'm showing the binding pose of Vasopressin within the V1AR binding pocket. And as I already mentioned, Vasopressin interacts with certain key residues. And on the right here, the mutation data from a binding assay. And we can see here some of the residues such as the lysine 128 and glutamine 131, both makes hydrogen bond with the compound, but when lysine 128 and glutamine 131 were mutated to alanine, for lysine 128, the affinity towards the ligand were significantly decreased. Similarly, some of those aromatic residues were when mutated to alanine, we observed similar results with decrease in affinity. And this suggests that these residues are extremely important for ligand binding. And this makes sense because this compound is a competitive inhibitor of ATP. And there is published literature suggesting that these residues are important for ATP recognition. So this compound, as it competitively binds, it binds to the similar location, replacing ATP, and keeps the receptor at an inactive conformation. Then we also determined another structure of V1AR dimer that is treated with conifeptin at 3.5 angstrom. And we got similar results with two protomers, little bit of nanobody 6 density, and surrounded by lipid-like densities. However, in this structure, we couldn't see anything protruding upwards or that could correspond to ECL2. Now, if you remember, for the conifeptin in the monomer complex as well, we couldn't see any ECL2 densities. We saw something at some density spanning the binding pocket on top of the V1AR binding pocket, and that density could belong to conifeptin, but we are not sure at this point. It could be ECL2 as well, but we tried fitting the ECL2 in that density. We couldn't. So nevertheless, we tried fitting the ligand, and the ligand fits there. And this is how the ligand sits on top of the binding pocket into that density. Then I overlaid our Vasopressin and conifeptin treated structures together. I'm just showing one protomer for the ease of visualization. And we can see how these two ligands occupy the binding pocket. While Vasopressin buries deep within the autosteric binding pocket, conifeptin occupies the top of the binding pocket closer to the extracellular side. Then there are certain interactions that are common in both the structures, which is the lysine 128, tyrosine 216, and glutamine 311. And these residues were tested in mutation assays for Vasopressin, and I assume they will have similar effects for conifeptin as well. As again mentioning, even though it's a non-selective antagonist, it's a competitive inhibitor of ATP. So it's not surprising that the conifeptin interacts with these key residues required for ligand recognition. There are some interactions that were not shared between velcovaptan and conivaptan, and some of which are listed here. But I would like to draw your attention towards this toggle switch residue, tryptophan 304. Velcovaptan interacts with this one, but conivaptan doesn't. And that might suggest the differences in antagonism pattern between two different compounds. Again, when we overlay the structures of both the protomers, we can see here, even though it's the same receptor, same purification, just the ligand was different, the protomers do not align perfectly. This suggests that there might be some structural differences or conformational variation present between the two structures, which could be driven by the ligand binding. Nevertheless, the dimer interface in our structures are primarily mediated by TM1 and is packed with hydrophobic interactions. So, as a summary, we have got a TM1 mediated dimer interface. Velcovaptan is buried deep within the pocket while conivaptan occupies the top of the pocket. And ECL2 adopts a lifted conformation in velcovaptan, whereas we couldn't see any ECL2 density in my conivaptan treated dimer. And velcovaptan interacts with the conjugate toggle switch when conivaptan doesn't. Then, while preparing this thesis at the end of my PhD project, another group came up with the V1AR structures at inactive state that is published literally in January this year. I was devastated, mostly. But they have got four structures, one at ACO and three with three compound bond structures. So I had to look at them quite closely. Therefore, here I am showing the comparison of our dimers with their APO structure. For the velcovaptan here I'm showing the superimposed structures of velcovaptan bound dimer and their APO structure. And the most striking difference observed in ECL2 conformation. In our structure, ECL2 adopts a lifted conformation, no surprise, because velcovaptan occupies the binding pocket and replaces ECL2. While in their APO structure, ECL2 acts as a lead on top of the binding pocket. Another striking thing is, it's quite interesting because we can observe in their ECL2, it adopts mostly like an alpha helical conformation. However, when it moves upward, it changes its conformation, it's mostly beta sheets. They didn't look deep into that, but just an observation. Then we again compared our conivaptan bound V1AR with the APO V1AR. Now, this is a tricky one. Because here we can see they have modeled ECL2 there and we have modeled conivaptan there. And when we overlaid those two structures, they clash. Then we compared the maps side by side. The pink map here is of our structure and the yellow map is the published one structures. So if we look at those densities, the densities look significantly different. In the density that they have modeled ECL2, that looks like that density belongs to ECL2. Whereas in this density here, we couldn't fit the entire 25 amino acids of the ECL2. But when we fit the ligand there, it fits to somewhat extent. So also there is differences in arrangement of the TM helices as well. So what we have hypothesized is that our map from conivaptan treated sample should be interpreted with caution, because the density in the pocket might be conivaptan bound near the extracellular entrance of the V1A ligand binding pocket, and that might destabilize ECL2, and therefore we couldn't see a well-resolved ECL2. However, the density in the pocket is ECL2 and conivaptan is not stably bound, or the density in the pocket is an average with some particles containing conivaptan or some particles of ECL2. At this point, we cannot conclude this claim. Then we compared our V1AR dimers with another of their V1AR dimer, which is the bellovaptan bound dimer. The reason I chose this because bellovaptan belongs to the same class of antagonist surfactants as velcovaptan and conivaptan. So here I'm showing the superimposed structures of both conivaptan, velcovaptan, and bellovaptan bound structure, and the most obvious differences in the ligand binding force. While velcovaptan and bellovaptan occupies deep into the binding pocket, conivaptan doesn't. and Vasopressin and Vasopressin appears to be adopting a similar binding force inside the binding pocket. Then we also compared our dimer interface with this. Then our dimer interface is predominantly mediated by TM1, whether in their case it's mediated by some parts of TM1 and some parts of TM2 and TM7 as well. That again suggests that structural differences are there, even though it's the same receptor, which could be driven due to different ligands. Now, this brings me to my second major aim of my PhD, which was to determine the active state V1AR structure. So, as Alok already mentioned before, I also utilized and optimized different strategies using dominant negative GQ protein, the mini GQ, but I couldn't get a stable complex until I used the G protein mimic, which was the savior. And I determined this expressed and purified using the protocols that I already described before. And we could see a complex in SEC and all the corresponding components that are present in the complex, suggesting a successful complex formation. Then we took this sample to Cryo-EM and we obtained the structure at 3.25 angstrom. Here we can see in our structure both G proteins bound, nanobody 6 bound, and in the side cutout here I'm showing the ECL2, how the ligand is occupied, and how G alpha 5 helix of G protein is inserting into the intracellular crevice of the receptor. Now here I'm showing the ligand density and how the ligand is positioned in the binding pocket. And yeah, as I already mentioned, AVP has two different parts structurally, the C-tail and the C-tail at the top and the cyclic ring. So in our structure, the cyclic ring of AVP occupies the deep within the binding pocket, whether the C-tail moves upward toward the extracellular domain. So for the AVP residues 1 to 6 form extensive interactions with receptors deep in the TM domain. We mutated a number of these residues that interact with AVP to alanine. And here I'm showing the affinity values for AVP determination on right. And alanine mutations of these key interacting residues result in significant decrease in affinity. And this result was again reflected in their ability of these mutants of the receptors to activate the receptor with a significant loss of potency for all the mutants. However, the maximal response was variable for some of these mutants. Then here I'm showing the interactions of the C-tail and the C-terminal 3 amino acids of the peptide extend, as I already mentioned, extend towards the extracellular phase, and particularly interacts at the top of the TM1 and the N-terminus and TM7. Again, we mutated some of these residues to alanine, and here I'm showing the loss in affinity. And these results were further reflected in the functional assays. So for alanine mutations of key interaction with the residues in TM1 and N-terminus and ECL3 that interact with the extreme C-terminus of this peptide are crucial for affinity, but that might not the same for potency, and they have variable responses to Emax as well. Now, we also modeled, we have a putative ion density near TM1, TM2, and near the ligand, and we modeled magnesium into that density because there is evidence in literature that AVP binding affinity increases with magnesium ion, and also in the oxytocin receptor structure, at a similar location, magnesium is mapped. And look, since these receptors are structurally quite similar, we think that this could be magnesium. Here I'm showing the interaction between the G-alpha-5 helix and the receptor intracellular cavity. And two interactions that were quite interesting to me was the glutamine 162 and 161 from the ICL2 that interacts with the alpha N helix of the G protein. So, as a summary of this active state structure, the cyclic ring of APP occupies deep within the binding pocket, whereas the C terminus engages towards the extracellular surface. APP makes extensive interactions with the binding pocket key residues, consistent with mutagenesis and functional studies, as well as our published literature. Interactions of the cyclic ring with B1AR are all required for APP affinity. And APP C-terminal tail interactions with the TM1 and N-terminus are essential for affinity, but ECL3 interactions are important for both affinity as well as the agonism or activation of the receptor. We also modeled the putative magnesium density near TM2 and APP, positioned between the two acidic residues. Now that we have both our active and inactive state structures, our main goal was to understand what drives the activation and inhibition of vasopressin receptors. So we compared our active V1AR with inactive V1AR structure. Here I have overlaid the two structures and showing and I'm showing here the TM arrangement in the extracellular side and the TM helices arrangement on the intracellular side. And we can clearly see on the intracellular side how TM6 is moving outward in the active state structures. And we have almost 5.3 angstrom TM6 outward movement in our active V1AR compared to inactive V1AR. Then we again observe differences in some of the conserved residues. For example, if we look at this toggle switch motif, the toggle switch residue undergoes significant conformational rearrangement in the active and inactive structures, including this fifth motif, the tyrosine in the active state moves quite inward. Then again, in the DRY motif, the arginine undergoes significant conformational rearrangement. And if we look at this tyrosine in the NPXXY motif, we can see that it goes further away from the compared to the inactive state residue. So all of these suggest that these activation microswitches, they drive activation of the receptor, and we can see the comparable differences between active and inactive states. This brings me to my third aim, inactive state V2R structure determination. So similar approach was obtained with construct design and AlphaFold predictions, and I have highlighted the region where fusion was made, and also the TM7. We have got similar results with differences in confidence score. Then all these constructs were again tested in a similar way in the Brett assay, and out of the 9, only 5 of the constructs gave us robust nanobody 6 engagement. And here I'm showing in highlighted boxes the construct that gave us good nanobody 6 engagement compared to the others that didn't. Among all these, V2R Chimera 1 was predicted with the highest AlphaFold confidence score that showed us the biggest nanobody 6 binding in pharmacology. So this construct was taken for structural analysis. And I again tested this particular construct, Chimera 1, in presence of different compounds for its ability to bind different compound. And we have got similar profile with increasing antagonist concentration, inhibition of nanobody 6 dissociation. Then Chimera 1 was expressed and purified in presence of the compound tolvaptan, and we have got a monomer fraction, unlike V1AR. And however, we couldn't get a high resolution structure. Even though this construct showed robust nanobody 6 engagement, in biochemistry, we have a beautiful complex, but in Cryo-EM, it suffered through lots of different issues. I'm not going to discuss those. Nevertheless, when we were trying to optimize this, another paper came out with the virtual structure with the same ligand. So we decided not to pursue this. As a conclusion, we have designed a coordinated strategy including AI, pharmacology, and structural biology to determine inactive structures of GPCRs. And we designed a bread-based sensor for studying GPCR activation and inhibition and characterized V1AR in both active and inactive conformations bound to peptide and non-peptide ligands. So yeah, for future perspective, I think we'll validate the functional relevance of V1A dimer assay using saturation breadth. I have already designed the constructs and also determined V1A homodimers bound to different antagonists. And there is evidence in literature that this receptor forms heterodimers with other receptors within the family and outside the family. So I would really love to explore that area. And with that, I would like to it's time for acknowledgement. I would like to thank my supervisors, Denise, Patrick, Brian, Matt, Sam. Denise, of course, my God. So it's an honor. I would have not been here. When I was in Arun's lab, I got introduced into GPCR structural biology and I got really interested. And then he asked me, okay, so what are your plans? I'm like, okay, I want to do PhD. Then it was my PhD interview and he asked me, who else is on your panel? I'm like, Denise. He's like, what? You are giving presentation to Denise? You gotta give your good game, mate. No. So I know how you need to give your best. And I was under so much pressure. I couldn't sleep that night, and it was India, Australia, the timing difference. And nevertheless, I got in. And before coming into this lab, I was following Denise's work, Patrick's as well. Like so closely. So it was really an honor when I got to work with them. It's amazing supervision, and just I couldn't have done it without them. And I would also like to thank Brian and Matt and Sam. Always annoy them, send them texts. And with any inconvenience in data processing, biochemistry, or beat pharmacology, Sam literally taught me how to do a simple graph thingy like 2-3 days before my presentation. So it was really, I'm really blessed to have you guys as my supervisor. And George, this would have been not possible. I have cloned constructs closer to almost 100 throughout my PhD project in different formats. And George gave cloning classes to Alok and me together. And it was amazing. Thank you so much for that. And I would also like to thank our center managers, Jackie and Tracy, for managing everything. And Tracy, thank you. You have helped me enough in all the way possible. And when I was in US, Tracy took care of everything because I got COVID in 3 different continents, in Asia, in Australia, and in US. So yeah, basically. So Tracy was amazing. And I would also like to thank my parents for believing in me. Because from where I come from, people are not aware of GPCRs, let alone Cryo-EM. So yeah, and I would like to thank my friends. Hello, we rant all the time. We were texting yesterday. I don't know, 5 in the morning, we didn't sleep. And Favika, thank you so much for always being there. Nancy, Meow Meow. And I would like to thank all the present and past members of the MRPE lab for your continued support. I would also like to thank my panel members, Nick, Charlie, and Nat, for always supporting me throughout my milestones. And I would like to thank of course, the center for accepting me and giving me this opportunity to do this PhD. And I would not last the least, I would like to thank my mentors in Pfizer. I had an amazing time there. They were really welcoming. I was a little bit skeptic before going to US, but that group is just amazing. It was just so nice. And I would like to thank Mark Whitley and his work at the University of Birmingham, from where we got all those mutation data for our compound bound and AVP bound data. And yeah, nevertheless, yeah, now it's time to do the real talk. I'm looking for some uh, positions. If you or any of your colleagues know of anyone who's hiring, please let me know. I would love to reach out to them. Uh, and yeah, thank you for listening to me and thank you for your time. I'm happy to take any questions if you have.
All right. So any questions from either online or in the room from Minakshi? Arista?
I was wondering with your nanobots assay, if you ever tried antagonists just by themselves to see whether they would increase the basal nanobotics and you got any?
I did. For V1AR, we couldn't see any inverse agonism. These antagonist compounds directly blocks it, like smash it. But for V2R, for some of the compounds, I didn't show this data here. Some of the compounds we could see slight increase in inverse agonism. So it was there. It did increase the nanobody-6 binding when it was antagonist by itself.
And it says that at a different chimeric
Yes.
Because potentially some of the ones that would show I don't know, less nanobody basal would give you a bigger range for recruitment.
Yes, 100%. That was the whole point because constructs that were predicted with higher confidence score by AlphaFold actually showed a better nanobody-6 engagement. And it's not only in our V1AR case, it was the same results in our V2R project as well. So now we have data from N of 2, if we want to say it like that. That strategy, that pipeline, it works.
So I guess Aristo's question is when you tested those chimeras inverse agonisms, did the ones that have lower basal brand have a greater increase in nanobody-6 binding? I don't think they did.
They not necessarily. It was just an inverse agonism, but not necessarily where a greater nanobody-6 engagement was observed.
And I guess the best chimera that you got, what was actually insertion on because you said chimera 4.
Yeah.
Was it more resonance of the 4? Was it?
That's in my. Oh, I forgot to talk about my Pfizer stuff. I'll come to that.